Today, we will delve into the fascinating field of pharmacology, specifically focusing on the intricate world of epigenetic regulation. Epigenetics refers to the study of heritable changes in gene expression that do not involve alterations in the underlying DNA sequence. These modifications have emerged as key players in numerous physiological and pathological processes, offering novel avenues for therapeutic interventions. In our discussion today, we will explore various aspects of epigenetic regulation and its potential applications in medicine. By inhibiting the activity of histone deacetylases, DACs, DAC inhibitors, HDAC, promote the acetylation of histones, which leads to the activation of gene transcription. This can alter the expression of genes involved in cell proliferation, differentiation, and apoptosis, making HDAC potential treatments for cancer due to the frequent presence of aberrant epigenetic modifications in cancer cells. DNA methylation is another crucial epigenetic modification involved in gene regulation. The addition of a methyl group to cytosine residues in CpG denucleotides by DNA methyltransferases, DNMTs, represses gene transcription. Aberrant DNA methylation patterns are observed in various cancers, contributing to tumor progression. DNA methylation inhibitors, such as 5-azacitidine and decitabine, block DNMT activity and promote DNA demethylation, leading to the reactivation of tumor suppressor genes. These inhibitors have shown promising results in the treatment of hematological malignancies. Histone proteins form the structural backbone of chromatin, and their post-translational modifications play a critical role in epigenetic regulation. These modifications, including acetylation, methylation, phosphorylation, and ubiquitination, create a dynamic and complex histone code that determines gene expression patterns. For example, histone acetylation is associated with transcriptional activation, while histone methylation can either activate or repress gene transcription depending on the specific site and degree of methylation. Understanding the precise role of histone modifications is vital for deciphering epigenetic mechanisms and developing targeted therapies. Stem cells possess remarkable self-renewal and pluripotent capabilities, making them invaluable for regenerative medicine and tissue engineering. Epigenetic modifications play a crucial role in maintaining the stemness of these cells as well as guiding their differentiation into specific lineages. By manipulating epigenetic marks, researchers can enhance stem cell expansion or direct their differentiation toward desired cell types. This ability holds immense potential for therapeutic applications in tissue regeneration and repair. Neurodegenerative diseases, such as Alzheimer's and Parkinson's disease, are characterized by the progressive loss of neural function. Emerging evidence highlights the involvement of epigenetic dysregulation in the pathogenesis of these diseases. By targeting specific epigenetic modifications, such as DNA methylation and histone acetylation, it may be possible to modulate gene expression patterns and halt disease progression. Epigenetic drugs could potentially offer new avenues for neuroprotective interventions and disease-modifying therapeutics. Apart from DNA and histone modifications, non-coding RNAs have also emerged as critical regulators of gene expression. Small non-coding RNAs, such as microRNAs and long non-coding RNAs, can modulate gene expression by binding to mRNA transcripts and influencing their stability or translation efficiency. Harnessing the power of RNA-based therapeutics holds great promise in manipulating the epigenetic landscape for therapeutic purposes. Techniques such as RNA interference, RNAi, and antisense oligonucleotide therapy offer exciting possibilities for targeting specific genes implicated in diseases. Although epigenetic therapies hold immense potential, several challenges need to be overcome for their successful development. One major hurdle is the specificity of these drugs, as off-target effects can lead to unwanted side effects. Another challenge lies in efficient delivery of epigenetic drugs to their target tissues or cells. Additionally, the heterogeneity of epigenetic modifications across different diseases poses a challenge in identifying optimal targets for therapeutic interventions. Addressing these challenges will require collaborative efforts from researchers, clinicians, and pharmaceutical companies. Non-coding RNAs, once considered junk molecules, have now emerged as key players in epigenetic regulation. MicroRNAs, for instance, can bind to target mRNA transcripts, leading to their degradation or translational repression. Long non-coding RNAs, on the other hand, can interact with chromatin and modulate gene expression by recruiting epigenetic regulatory complexes. Understanding the intricate interplay between non-coding RNAs and the epigenetic landscape opens up new possibilities for therapeutic interventions. Epigenetic modifications have a significant impact on the regulation of immune system function. Alterations in DNA methylation or histone modifications can influence immune cell differentiation, activation, and effector functions. Dysregulation of these epigenetic marks may contribute to the development of autoimmune diseases, immunodeficiency disorders, and inflammatory conditions. Targeting epigenetic modifications in immune cells holds promise for therapeutic interventions, such as modulating immune responses or enhancing cancer immunotherapy. Identifying reliable biomarkers that can predict patient response to epigenetic drugs is crucial for personalized medicine. Consequently, considerable research effort is directed toward the discovery and validation of biomarkers that can assess treatment efficacy and guide therapeutic decisions. Biomarkers may include DNA methylation patterns, histone modification profiles, or specific non-coding RNA expression signatures. Integrating biomarker-based approaches into clinical trials can refine patient selection criteria and enhance treatment outcomes. Emerging evidence suggests that epigenetic modifications play a crucial role in the development and progression of cardiovascular diseases. Aberrant DNA methylation, histone modifications, and non-coding RNA expression have been implicated in conditions such as atherosclerosis, hypertension, and heart failure. Understanding the epigenetic mechanisms underlying cardiovascular diseases may offer new therapeutic targets and strategies for preventing or treating these prevalent conditions. Epigenetic reprogramming involves the erasure and establishment of epigenetic marks, potentially allowing the reversal of aberrant epigenetic modifications associated with diseases. This approach holds promise in cancer therapy, as it offers the possibility of resetting the epigenetic landscape in malignant cells and restoring normal gene expression patterns. Epigenetic reprogramming techniques, including the use of small molecules or genetic manipulation, may pave the way for innovative approaches in cancer treatment. In sum, exploring epigenetic modifications reveals valuable insights into how genes are expressed and diseases develop. Manipulating DNA methylation, histone modifications, and non-coding RNAs offers numerous therapeutic prospects spanning oncology, neurology, regenerative medicine, and immunology. However, more comprehensive investigation, partnerships, and optimization are essential to fully harness the power of epigenetic regulation for enhancing human well-being and combating ailments.